Okay,
I don't want to be recorded.
we're
No.
recording now. Okay, hello everybody. Welcome to the end of Textile Summer 2023. This is an opportunity for the three of us to answer questions that you all have been submitting with your modules in a little bit more of a discussion format, so you can kind of see how we think about things as researchers, how researchers might think about things slightly differently from one another, and just another opportunity for you to get like insight into some of the greatest scientific minds of this generation.
Actually communicate with each other.
Yeah,
Actually,
and
we
then we do
actually
talk.
hang out.
Did they not think we did?
I feel like since we teach our modules
No.
individually,
True.
uh, yeah, we actually hang
That
out.
we work together and we collaborate, except Nels doesn't want to collaborate with me. That's why I want you to message me about your project so I can be
Well, I don't collaborate with Phuong yet either,
Not
so
yet, no.
I have
yeah.
not processed any of her videos for her. So,
Food and I have collaborated. Food, I have been at Hopkins in person for one day, and I have already been mentioning your name in extracellular vesicle work. So
Oh my gosh, getting my name out there and EVU.
I'm going to Hopkins.
Totally.
We do communicate. We are lab members. We did share offices for quite a long time until I
Somebody
moved
stole
away.
Holly's desk.
And now
That-
sits at
uh,
my old desk.
Now it's
So
just like success now.
I will start us off with some of the questions from earlier in the program because they're a little bit broader. So the question that was asked is, with so many possible areas of research to consider, how do researchers pin down what exactly they'll run their next experiment on? So I think we can I'll start. I think you can answer this on a few levels. So when you yourself are a trainee, oftentimes what you're brought in on is whatever grant your principal investigator has. So when you first start, you're going to choose a research area or an experiment that has funding, and it's a pretty easy decision from there. Then once I was in my PhD and I knew what my grant was on. Nels and I were on the same grant for quite a while, which is actually quite flexible. It's like the harnessing the data, the revolution grant. So as long as we were using data science, we could pretty much do whatever. And so when we are doing that, I have like 100 ideas for experiments. Because I was just a student, Elizabeth helped me narrow those down. I typically would try a few of them, and whatever ones were working out, that's what I move forward with. I know this approach is very different from Phuong, so I think I'll throw it to Phuong next. How do you decide what to work on next, Phuong?
Hmm. How do I decide what to work on next? I mean, I think you are correct in that in terms of grad school, it's just really dependent on what types of funding is available to your lab. So I will say that I didn't come in thinking I would work with extracellular vesicles, but here we are. And that was just what Elizabeth had available in terms of funding at the time.
Mm.
But to be honest, I do feel like a lot of it is I do feel like in grad school, and this is the same for undergrads and high school students, we're still students, you know, and I think that I didn't come in with a perfect idea of what I wanted to do anyway.
Mm.
Uh, so being open-minded, and even if it's a project that's given to you, I think you learn to make it your own,
Mm.
for sure. And and thankfully our PI has been super supportive in taking this project in the direction that I'm more interested in compared to what my predecessor was more interested in. So I've definitely taken like my own spin on this project, even though it wasn't like anything that I didn't come in thinking that this was the perfect project. I didn't even think I would be doing my project, honestly. But you can definitely have opportunities to make it your own. And yeah, I'm not sure if that answers your question, but
I think it gives a perspective. And Nels, is yours different than ours?
I not a ton different. I think only thing I would add is I think you should like I think one important thing is thinking about what projects interest you the most, because like so many potential collaborations or ideas are going to pop up.
also maybe pop off.
Sometimes projects pop off too. But I think
Sometimes they'll go really poorly.
also that.
That too.
But I think it's really important to be like constantly checking in with yourself, be like, okay, like is this an avenue I really want to put a lot of time into, or is there a new idea that I could also do? And then talking with your mentors about it. So for example, I was brought in to do specifically machine learning applied to MPT, which is really what I talked about. Like
Oh, was I?
what?
I was too, and I did not do it.
True. And so I'm still gonna do that, but one thing that one of my collaborators, Brendan and I have noticed is that the data quality control pipeline from wet lab to machine learning is seeming to add a lot of noise in the end. And so I've got really passionate about, okay, like how can I build software tools that we can use for our current data, but also future data collection and improve data quality to increase the signal and reduce noise. And so I think within whatever like your main project is, there are ways to find other smaller things that really interest you that you can put time into. So I think just constantly reflecting and being like, this, do I want to put time in this? There's so many things, you can't put time into everything. So really finding the few things you're really passionate about and really diving deep into those is important.
I think Nels brings up a good point. So Nels and I are both computational researchers in a wet lab. Both of us have done some wet lab work, but we both approach it very differently. So on the computational side of things, I'm very interested in data science quickly. I'm just interested genuinely in like studying data. We've laughed about this in that I will use Excel rather than automating something like manually if I could just study data and get like a biological result for microglia faster. Nels, as you just pointed out, is very much more into like the software development side of things. And I find that fine, but I would much rather be like staring at cells all day. And that's totally fine. That shows like another way that you can like bring your own personal interest in. And it also reflects our career goals because Nels wants to be a data scientist, right? Unless it's changed.
Yeah.
And I want to be a professor who has some software people and who has some wet lab people. And so that's totally fine too. Yeah, I think that's good. Okay, here's one that is more experimental. So the question is, I'm still a little confused about what experimental methodology means. And this question also came before Phuong's experimental module, so just keep that in mind. So the person continues to say, I Googled the term, and what I understand, it's a process where a scientist tests their hypotheses by manipulating many variables. So I'm guessing that the different methods are just steps in the process of carrying out your experiment. I would say people can refer to experimental differently. So some people just call anything that we consider a wet lab to be an experimental method. And I think they would probably bulk method into like a larger process. I think maybe Phuong will agree. Like I think I would call PCR a method, but not the individual steps of a polymerase chain reaction methods, even if they are the methodology of the method. Computationally, machine learning is made up of a bunch of different algorithmic approaches. You could call those your method if you wanted to. I use image processing. That's my method. I don't think it is so cut and dry. And I think people will refer to it as a bunch of different ways. But yeah, it's any large set of steps or processing you do to obtain experimental data.
Yeah, I would say the same. And I think it's important to differentiate methodology from like the different parameters that you're using in your experiment, I guess.
We
so
would
like
call that optimized.
that is I call that optimizing. Yes. Absolutely. So like I guess for example, if you're running like, I don't know, yeah, since we're on this topic, and since this has been the bane of my project for the past year, if you're running like a PCR, almost 2 years.
Mm-hmm.
Right. Wow. It's been a while.
Anyhow, don't make her go through
anyway, if you're running the PCR, right, I would call PCR itself as a method, and the steps in PCR would be like the steps of the PCR. And then in the PCR, what you do is that you run it and then you check to see if your results, if you get any results, and if they're interpretable. If they're not, you're going to have to go back and change a gazillion different things. You might want to change the temperature of your reaction. You might want to change the different reagents that you use. You might want to change the like instrument that you're using or the different fluorescent dyes that you're using. So there's a lot of different parameters that you change. And then once you've changed them, you got to retest, you got to rerun the experiment, right? So I would say that I wouldn't personally consider that as a method. That would be more optimization. Like when I go in and I change the different experimental parameters, that's more optimization rather than like a method.
What they may be referring to even as experimental variables is like when we have variables in our animal studies, we have something that's like a non-treated control, and then we have like a therapeutic treatment and an injury. I wouldn't call those methods. How we make the injured group, that's a method, but those three are experimental design. And so those go into what are you actually testing for and what's your hypotheses. A good way, I guess, to understand methods is if you go into research papers and you see the methods section, those methods sections are going to be split into subsections, which are typically a much larger method. So if you go through those, you can begin to understand what scientists consider method and what scientists consider steps and optimization towards that method. Like computationally, you could say like a neural network is the method if that's what you're really like focusing on. But if you're just using someone else's neural network, you might say it's just a step in like your larger process. Now, do you agree? I
would agree with that. Yeah. I don't think I have anything more. I think y'all covered it.
So I think that's a good question, and that I haven't thought about it in a long time. Probably since like early college when we had to like define those things. But it's really going to depend on your field. I'll keep going just because I'm quite general. So when publishing a research paper, do you publish a data management plan along with it? And how do readers access an author's data management plan? So I think that's a really fantastic question, as this is like a big discussion in a lot of fields right now. Some fields, yes, some fields, no. The NIH does require you on grants now to submit data management plans. The NSF might as well, but I don't remember. The NIH being the National Institutes of Health. And then on publications, depending on the journal, sometimes you have to say a data management plan. Typically, by the time you publish, they're less interested in how you manage the data and more interested in where the data currently is now and if it's accessible. The only, I would say, points when it's important how the data was managed if you're working with like human subjects, or if mismanaging computational data or analysis might change results or something like that.
Nels is doing a lot of data management and storage right now.
No.
It's important to do it right.
But doing it right is hard. And there's a lot of debate about what does right mean and what is sustainable and what is like green and economic. And so if you really want to see data management plans, I would go to like grants.gov and look up some NIH grants and look at what those data management plans look like. And there's a lot of literature on data management plans as well in bio and computational work. So you can actually read papers about data management if you want. Cool. Next one. Phuong. Here, this will be for you. We can both answer it, but I think Phuong should start. I wonder how researchers modify and change their experiments throughout. How do you document that? And what research do you have to do before you design an experiment?
Great question. I'm pretty sure you just asked. I think the answer to this question is really like, how do you do science? Because I feel like these are all very important steps in being a researcher. And I think this also touches upon data management, like what we were talking about before, in terms of keeping track of like your data and like what you've done. But I would start with the very first step is to have a question. That's important. You got to start with a question. And then, okay, you got to start with a question, Hawley.
Wait, I'll be right back.
Run around.
You start with a question.
Wait, Phuong, wait. Michael's had a sale for teachers the other day.
For what?
Michael's had a sale for teachers, and I needed some art for my apartment. If I can open this and people cannot be too annoyed with me online, I can help you out in just 1 second.
Oh my goodness.
Genuinely, this is art for my, yeah, it's the scientific method.
Okay. It's gonna be like a poster. Oh my God, it actually is the scientific.
Other side is The other side is lab safety. I'm genuinely putting these on my wall.
Michael,
Hi,
so
Mimi. Okay, so first,
Michael,
Just
so
for
please.
teachers.
For teachers.
you might observe something. This is in reverse, but it says make observations.
Okay, yeah, that's true. Okay, before you have a question, you have to make an observation of which you then ask the question about the
And then you
observation.
ask a question. This is it
There we
inverse.
go.
And then
And
you
then you formulate a hypothesis. And I will say that hypothesis is should be based off of what other people have. You know, you go and you do research, you go read papers, you go online, see what other people have done
Here we
to
go.
address it.
Yeah.
And then once you've read about what other people have done, then you create your own experiment. Then you can start drafting your own protocols and start thinking about your experimental design. And then you run the experiment, and then you get results, and you analyze those results, and then you store it and you manage your data.
I would disagree with this one. And I would say after you analyze your results, you go, is it wrong? I need another
And you
experiment.
go back.
Yeah. Is it at the point where I'm ready to publish a paper yet? Did I find something interesting? Did I reach my hypothesis? I think you do that until your PI thinks that you're ready. And then you report a conclusion.
Absolutely. Most of your time is going to be spent in experiment and analyze data for sure. You just go back and forth, honestly. It's a loop. It should be a loop, honestly. Yeah.
We have lab notebooks. You learn to keep a good lab notebook and you say, on this day I used this, on this day I used that. If the results are weird, you go, did I do something weird? If the results look good, then you do that again and see if they still look good. And you learn more about like advanced design and you can do like multivariable things. Oh, Nels turned hello? Nels?
We need to move.
One
1 second.
Give us a second.
Okay, I'll talk to myself. And you learn more things. But yeah, this was on sale at Michael's, and that genuinely is what we do. Sometimes I think part of the question was, how much research do you do? I think for me, along the way, I'm doing research and I come up with some conclusion, and then I do more research to write the paper. And then oftentimes in that process, I've thought of new experimental questions. So it's not necessarily that I had to do a lot of research to ask a question. It's typically I had to do a lot of research to figure out why stuff's going wrong, and then research to write the papers. And in that process, you have a new question. Hi. We'll get to the next one. Okay. Now mine are getting topic specific. So we can either hop to Phuong and then back to me. Do you want to do that?
Sure. Yeah, I have some general questions that we can go over.
Cool.
Somebody asks, okay, there's biological replicates and technical replicates. What's the difference? And that's a great question because honestly, even I get confused sometimes. Yeah, it's important in science to show reproducibility. Other people should be able to reproduce your result. And so because of that, and you also have to show that your result is significant. So because of these things, you are often running the same experiment multiple times. And you have a lot of different samples.
Many times.
And you have a lot of samples too. So I would say that it's important to be able to have a lot of replicates so that you can show that whatever you're seeing, whatever observation that whatever result you have is robust and it's like
and what we mean by robust is that there are statistical tests you can run. We use some nice statistical software, and that tells you if there is significance, and significance is like is a way to measure robustness.
Yes. So there's two different types of, like the person mentioned, there's biological replicates, which means that you have different. If you have a better way of explaining this, then let me know.
I always forget this.
So biological replicate, when we do animal studies, a biological replicate, a way we do it is to do litter matched animals of the same sex. And 3 of those would be biological replicates of each other, that they're biologically different.
They're biologically different, yeah. So you'd use the same, it would be like the same species, but different individuals.
Animals. And then what we would call more of a technical replicate is like every time that we stain, every time that we take, so immunofluorescent imaging, we take more than one image, we stain more than one slice. Anytime with our like nanoparticles, like with Nels and multiple particle tracking, you do a technical replicate. So you're gonna measure 3 different sizes of the same sample. And so that's just to make sure that an individual measurement you have of something is not a false individual measurement. And then you can have a biological or an experimental replicate, and that maybe you make 3 of that material and then you test that all 3 times, or stuff like that. In my experiments, we have 3 of each sex, and then I have 3 slices from each brain of each sex that I stain, and then each region that I'm interested in and each sex, I take 3 images of each region. And those are my versions of technical replicates within a biological replicate. Is that like downward trend of 3s? That becomes a lot of images. But yeah, that can change.
Yeah, so I would say, like you were saying, a technical replicate is kind of within a biological replicate.
It's more of a test of whether a method of measurement is doing well.
Yeah.
More so than a test of whether a biological phenomena is occurring.
Right. Yeah. That's a good way of putting it.
And we say we use 3 animals. So 3 animals is like on the low side. If you do what's called a power analysis of how many animals you really need for something like or how many humans you would need to approve something in FDA, the numbers get significantly larger. And so part of doing humane research and responsible research is starting with the lowest numbers you can and not moving up to these larger numbers until you've shown robustness in early stage trials and experiments. Yeah.
Right. Yeah, that's a good question though. I still get confused of what they mean.
I think also it depends on what you're doing.
Also depends on what you're doing. Another general question that I received was, are there any extracurricular activities or classes or clubs that you think would be valuable for high school students who want to go into research?
Right. Just right. You know, if you're interested in science, you get all the science and you're interested in it, and you probably find things that are good for you. The thing I wish I had started doing sooner is writing. Whoever in your high school is telling you that if you do engineering, you don't have to write, because people were telling me that, you're wrong. It's so much writing. Even if you go into industry, there's so much writing. Just write. Just write a lot. The rest of it, you enjoy it. It'll be fine. But if you don't enjoy writing, do that.
Yeah, do you enjoy writing now?
I think you can get away with not enjoying writing.
Yeah, but you still have to do it.
I think you need to find that, like, what style, like writing style you like how you like to do the active writing. So for me, I am not good about like if I'm just like, all right, here's 4 hours of my time that I'm blocking off just to write. I don't do anything for 3.5 hours and then I write for half an hour. What I've done that's been more effective, which is actually Hawley kind of introducing to it, is
Pomodoro. Yeah.
Where I do it in like 30-minute increments, and for the first 25 minutes I'm like really writing, and then for 5 minutes take a break, and that just works a lot better for me because it forces me to get things out, like actual just like literal words on the paper. They're not necessarily good. I'm for sure going to edit them later, but at least my brain thoughts are spilling onto the page somewhere. Versus if I don't do that, it's like everything is in my head for 2.5 hours, and then I don't really remember anything. Then I'm like, oh crap, I should probably finish up this writing session. So I think I enjoy writing more when I'm doing Pomodoro, because I just get in the flow of writing versus, oh God, I have to think about what to write next, and then I'm bored. So
that's probably not the answer you expected. But I think if you're interested in science, you're probably doing science. And just let yourself do science. Like, don't put a ton of pressure on yourself to be like, oh, I'm interested in research, I need this specific extracurricular. At the high school level, you just need to have interest in science. That's it. Be interested, get into undergrad, get a little bit settled in your classes, apply for a research lab, you're fine. You don't need to worry about what I feel like is a very modern pressure to have already started in research labs in high school. I started like sophomore year and I still got into a program and I was really only doing 5 hours a week. For probably until my senior year when I had a grant to spend more time on it.
Yeah, I didn't do any like science related extracurriculars in all of
You
high
didn't?
school. No, I just played sports the whole time. But
Wow.
like I think
I
that
didn't do them either.
but like I knew that science was my favorite classes, so like I would really focus, like I put a lot of time to study and I would talk to my teachers about stuff, and I feel like I put a lot of time into it, but I don't think and I don't think if I had been doing, I don't know, started like a crazy biological club at my school, I don't honestly don't think it would have helped that much. Versus just like I wanted to enjoy high school and playing sports, and then it's like in college, okay, like now I can find this other
It
thing.
has a different perspective. I'll also add
Yeah.
to that because now that Nels points it out, I didn't have any science electives. I did science
Wow.
in school. I knew science would be what I did. I was in National Honor Society, which I thought was the most useless thing I had done. And then I was in art. Actually, I did a lot of art, which I think makes me good at data visualization nowadays. I was also in sports and choir. In
Wow, you're all
undergrad,
I play
in
orchestra,
undergrad,
so
wow, you're so
everybody.
skilled.
in undergrad, I also had I was in no science clubs. Now that I think about it, I wasn't even in student American Institute of Chemical Engineers. I was a tour guide. That's all I did. I did research. I was a tour guide. I did well in class. That's enough.
I also, yeah, I didn't do any like extra science clubs. I was doing research, but that was never like a huge. It was like no, I really enjoyed it, and it was a big part of my growth as a scientist. But I don't think there's always a need to do, like, everything you do doesn't need to be tailored towards science. I also think that like doing things that aren't science, one, less likely to get burnt out, but you develop other like really good skills. So like when I played
Yeah.
sports, I think I became a much better, just like able to interact with lots of people, able to lead teams. And so these skills that I think help me as a scientist were done from playing sports or playing an orchestra where you have to be just in sync with everybody. So I think you can do what you enjoy and still build those skills that are important to be a scientist, but they don't necessarily have to be in a science specific setting.
And then the final comment I'll make before we let Phuong tell you the exact opposite is I think that you're more interesting if you have hobbies that are not just science. Like don't optimize towards a career. Like just live your life. You're
Your
fine.
life.
Okay, now
Okay. As
Science
somebody
Olympiad,
who
president of Science Olympiad.
president of Science Olympiad, president of Science Club, president of Math Club.
Well, because
Yeah.
you weren't coordinated enough to play sports.
Okay, as somebody who did those things, very, very science heavy, I would say that what Nels and Hawley said, I completely agree with. I think I was boring.
No, I love you. I do. I don't
Thank
think
you.
you're boring.
I do wish I had other skills though, like music. What is it? Have you seen Napoleon Dynamite? That part where he was like, "Oh, I have crossbow skills." I forgot what he says, but it was really funny. Computer hacking skills, nunchuck skills. I also had other skills. And I think that would have been really great for my overall like mental health. But I will say at the end, senior year, I finally did something not science. And that was I founded the Harry Potter Club.
Ooh, this is on brand for you.
That's so on brand.
So that is not a science thing. I had fun. It was about magic. We talked about magic. And it was a good break from all the science I was doing.
Yeah.
And like Holly was saying, at that level, high school, honestly, like all the science clubs or like math clubs, I honestly don't think I gained that much from science and math club. You just kind of sit around and you
have
like
to
maybe you extract DNA from bananas, which is cool.
Mhm.
But I feel like I would have done better, or not better, but I think I would have gained more valuable lessons if I had learned like management or leadership, like what Nels was saying, through other clubs.
I also think you have to keep in mind, like, getting into research as a career is a pretty long trajectory. And the priority when you're in high school and in undergrad is to get a strong foundation in science. So that when you show up and you're doing research, now you're learning skills to apply science. Your main priority should just be learn science, enjoy life.
Enjoy life. Yes.
That's
I
it.
do want to emphasize though too, like if like I don't know, like robotics club or something like that, like if that's genuinely something you're passionate
Right.
about,
Yeah. If
like
you,
you absolutely should do
you
it.
should
I
do it.
think it's if you shouldn't feel like, oh, I know I want to go into mechanical engineering, so I have to spend 20
Right.
hours a
Yeah.
week doing robotics, but I hate it.
Yeah.
Like
Mhm.
Yeah.
for sure, if you're it's like science club, whatever, if you're passionate about, like do it, but just never feel like
I was
obligated. You should do it because you
passionate.
should do it because you really enjoy it, not because you think you should have to.
Yeah.
Yeah.
And I think Phuong really enjoyed Science Olympiad.
I did science. I loved
And
Science
Harry
Olympiad
Potter.
and
Some of
Harry
your some of your closest friends are still from Science Olympiad, but yeah, don't worry about it as like a stepping stone you need for your career. You'll be totally fine. Just do what you're interested in. Go where the cool kids are. Um,
Well,
I wasn't a cool kid, so I can't. I'm
was
having that one.
good at.
Okay, Phuong, what's your next
Maybe
question?
Nels was a cool kid.
my grade was like 40 people. Everybody was a cool kid.
Wow.
Yeah, that's a cool kid thing to do. No, that's not a cool kid thing to say. They would say some kids aren't cool.
Mm-hmm. And I think that's actually all the general questions I had. The other ones are more, well, actually, wait, no, there's another one. Um, maybe we already answered this. It
We can
was, it was, yeah, we already answered it. It was like, how do you create an experiment?
Oh, yeah.
That
A
was
lot
good.
of I also think I will add to a point to that. I have found that from a lot of feedback, not just from this summer, but the previous summer we've taught, the previous 2 summers I've taught, and then high schools and undergrads. A lot of people come in thinking they have to come up with their own research project. No, you will come into a lab that already does things. They'll have ideas. We'll ask you to explore within those ideas. But you're very young in your career. No, we're not expecting and I don't think you should expect like a Nobel Prize breakthrough from a high school project because most of those breakthroughs are like decades of work from like hundreds of people, but they just put 3 people on the award type of thing. Okay, I have, I guess, some more general ones. Some are about reading papers, which we got a few questions about. So when you write about what you read in research papers, I think this is specifically in reference to like reaching out to professors saying you're interested in the paper, but we can take it more broadly. How much do you write about what you've read? This person says, I usually do 2 sentences, but I've heard of people doing a whole paragraph too. So I just did all of my postdoc applications. I've had a few people messaging me about this. Legit, I wrote 1 sentence on each paper I was interested in. I was like, I really liked your 2020 paper on this. You're not proving to them that you can read their paper or you know their stuff better than them. All you're showing in that email is that you are interested in them enough to look up and read one of their papers. It's like crossing the box of, are you just sending the same email to everybody or are you genuinely interested in that project? And also telling them what part of their lab you're interested in. So if they do have a position or a PhD student who might want an undergrad or high school student, they can send you to them. Any input?
That's literally why I think Elizabeth was willing to take me into her lab when I was doing master's was because I looked at her paper list and I'd seen recent ones with machine learning. And
Mhm.
we wrote 1 sentence. I gave her a little bit of my background. I was like I really enjoyed reading the papers about machine learning applied to the brain extracellular matrix. That's
Mhm.
research. That's the kind of research I'm interested in. I didn't like be like, oh, this specific result was really cool, or
Yeah.
I have a new idea for a project based on this. It's just like I saw this paper, I read enough to know that it has machine learning and brains in it, um,
Mhm.
and I thought it was cool. And I think that's she knew. Oh, okay, he like didn't just randomly select me and send an
Mhm.
email to me. He at least looked at my website and saw some papers on there.
Right. Honestly, it's better that it's shorter. A lot
Mhm.
of people just don't have time for you to regurgitate their own paper to them,
Yeah.
They want to know why you emailed them and if they can do anything about it, and they want to be able to make a decision quickly. So,
Yeah.
brief introduction to who you are, why you like them, and what you hope to happen next. That's the best you're gonna. Don't go crazy. I will say there are other instances when you're reading a paper and you're writing about it that you write a lot more, such as a lot of PhD. Some of your first exams is can you critique a paper? So keep in mind, we're not critiquing papers really at all until the end of like our first years of our PhDs. And then we write 5 pages of like a critique about these papers. So you're not expected at your level to give these high level critiques of papers. In undergrad, you'll probably be asked to give some critique and some feedback of papers, but you're building up those skills. It takes a while to get there. Yeah, cool. I have another couple broad.
Oh, I actually have a general one too that I just saw.
Yeah, you go.
And I'm not sure if we actually talked about this during the data management one, but somebody asks, how do you record, like where do you record results? Do you do it on your computer? I like to take notes with my computer, but should I switch to a physical notebook?
Okay, so I would say for this, whatever you have to have written down for your lab is going to depend on what your lab wants. So like Elizabeth requires us to have handwritten lab notebooks for wet lab experimental methods. And those lab notebooks are supposed to stay in the lab and they stay with her after we leave. So I wouldn't switch or anything unless your lab has specific requirements. I think when Nels and I code, we do like a notebook version, and so I keep all old versions of notebooks and I can rerun anything. Most results end up on our computers or Google Drive, and for me that just depends on the methodology where I keep it and what information about it. Food?
Yeah, I agree. It's honestly just what your lab, I think.
As a high schooler and an undergrad, you shouldn't be making those decisions. Someone should tell you where you should write and store everything, and you should follow that.
Yeah. And in terms of taking notes for classes, just do whatever works for you. Honestly, you don't have to worry about switching your entire
I'll
process.
also admit I only took notes in undergrad so that I wouldn't fall asleep in lecture, and I never looked at them again, ever. And I threw them away as soon as undergrad was done. Some people still have theirs.
Oh, yep. Oh, you don't? All right. I'll pretend that I
Club
don't.
president
I'll
would have
pretend that I
theirs.
don't have
Good
these.
memories.
Look, sometimes I forget how to do, what is it? The ice table. Have you ever done the ice
Yeah,
table?
you can Google that.
Yeah, you just Google it. You look at your old notes.
Yeah, I looked at my own notes.
So funny. Yeah,
Ice
so
table, what's that?
if you worry about them for class, I do that. I think one of the most important things now that I'm further in my research, and so I think something that becomes important end of undergrad throughout your PhD is how do you begin to keep notes on papers you read? That's really complex. Like, how do you keep ideas that you've had straight? And how do you remember like how they connected and all that stuff? There's a lot of different approaches to that. A really good book about that is How to Take Smart Notes. I recommended it earlier. And that's when I would start saying, if you want to make a career out of those research projects, that's when it becomes really interesting to record notes and like solidify them. But yeah, okay. I have some. Okay, this is also about research papers. Say you read a professor's research paper and didn't understand it entirely, but that research paper is what's most interesting to you about the lab. Should you write about it in basic surface level terms or pick another paper? I think this goes back to the other one. I think you just write like the topics and what you're interested in. It would be so surprising to me if a high school student or an undergrad understood my paper in its entirety. And I also would not want you to email me a summary of my paper in its entirety
Yeah.
at all. Please don't. But I also do not think you should put pressure on yourself to understand papers in entirety. I don't even understand all the papers that came out of the Nance lab in their entirety. Sorry, Elizabeth, if you listen to this. But like, I don't know everyone's exact methods. I don't know all the exact MBT results. I know the gist. If I gave it a deeper read and a few more days, maybe that's another thing to point out. How long you should spend reading papers. For like those critiques, those 5-page critiques we do for our like exams, we spend 2 weeks reading one paper. That's how you get like an in-depth critique. You don't have time to spend 2 weeks reading one paper. I know Food has a pretty regular paper reading habit.
I try. It's been not good this summer, but
yeah. And I tend to read a lot of papers in bursts. And so sometimes all I'll read of a bunch of papers are abstracts, and sometimes they're like seminal or really important to my field. And then I'll reread those multiple times and I'll give them a few hours for the paper, especially if you're really trying to reuse a methodology or like cite someone. Yeah.
How are you in your paper reading?
I feel like a lot of when I'm reading papers is I have an idea for some kind of computational work or some discussant section that I want, and then it just searching on different routes of finding papers, finding the ones that seem interesting, very quickly skimming the ones to see if they are like relevant to me or not. Then I like try to like get like go through as many papers I can as fast as possible without reading them in depth to get like I'll go through 50 and I'll find 5 that are actually interesting to me. That's I'll choose those ones to read carefully. So I think that's another skill you can work on is just not you don't need to quickly understand what the paper actually is truly finding. You have to understand if it's useful to what you're interested in.
That's a big one. Yeah.
Otherwise, you're gonna waste so much time.
You waste a lot of time. Yeah.
There's so much stuff out there that like has keywords that will show up in a search for papers, but aren't actually what you care about, or are so old that they're outdated. So and then I think then I just kind of like spend a lot of time be like, okay, this because for me, at least for computational work, then it's being like, okay, like how do they talk about this code? Do they have open source code that I can tie to this? So I think it's really like picking the sections that you really want to deeply engage with.
Yeah, I also think at this point, I think probably at different points, like when you are applying for graduate school, when you're looking for an undergraduate research lab, and when I was applying for postdocs, what you'll probably more likely do is pick some people that you're interested in and then just read a bunch of papers from the same people. And so I think that is something I do more now. Now that I have a better understanding of who are the big players in my field, I'll just read all of their papers even if they're moving topics, because I kind of more understand the organic movement of how they've switched fields and how that relates to their older work. And that's okay too.
Yeah.
You should read all my papers just to give them clicks.
Memorize them.
Phuong and I have a paper together that I didn't make them read in textile. I should have.
Yeah. Well, offended, but
I think that when I picked the papers, it was before our paper came out. The paper
well, you should all read that paper and then email us.
The summaries. Wait, if anyone that watches this video emails Phuong and I a summary of our collaborative paper, we will love that is okay. We'll just love
I will actually read that one.
Yeah.
I'll actually read that.
It's in IJMS, International Journal of Molecular Sciences. Phuong is the first author. I'm second. I think I'm second author on that paper. So you can find it. There's not that many Phuong Nguyen's, Hawley Helmbrecht's combos in IJMS. There'll be more in the future. Okay, cool. We're getting close to time, so I'll just keep pushing. I have just a few more questions. So this one's more for me and Nels. There's a few questions about data science. So the first one is, it's become quite clear that I've got to get my Python and GitHub together for any research. Our take. I would say no. My take, and Nels can go against this, is that Python is currently the hot moment. And there are things that are taking over from Python. I would say currently right now, if you want to get coding fundamentals, you should learn Python because there's just so much out there about it. You don't even necessarily have to learn GitHub. That's totally okay. But that will likely change in a decade. What's more important is that you're understanding how to code and how to learn how to code and how to work within worlds that there is code. Like MATLAB used to be the thing not even that long ago. And
MATLAB.
It's still huge. Now, is that your take?
I disagree on the GitHub take. I think GitHub will probably, I don't think GitHub is going anywhere.
I think version control is
sure.
Yeah.
I think understanding how to use version control is really crucial. I think it's always been important. I think it's becoming even more important with the very positive advent of a big push for open source data science and software engineering in the science domain. So I think understanding version control, how to get other people's code, how to push your code, stuff like that is really, really key. I also don't think it's terribly difficult to get the basics. Like I probably use 4 commands for 95% of what I do. And then if I can't do something, I Google it, and then someone tells me, and then Stack Overflow tells me what to do. Just getting comfortable with the polling, the adding, committing, and pushing workflow, I think is good.
And getting really comfortable with looking up things you don't know and what seems legit and what doesn't seem legit. I think that's a huge part of coding literacy, is how to fix errors and adapt other people's code.
Yeah, I agree with your point too about, for your other point, that I think Python is good, but I think like if your lab's doing Python, yeah, you should definitely like learn Python because your lab's doing it. But I don't think if your lab's doing Julia or MATLAB or Java for whatever reason,
or R,
right, you should also like learn Python like just to learn Python. I think I think if you learn good programming habits and skills, that'll always translate. Syntax is really easy to learn. Good programming skills are much harder to learn. So like my first time programming was actually in Java. And like I could not compile Java code right now because I don't remember the syntax, but so much of what I learned in those classes has enabled me to write better Python code just because I understand how to structure code at a high level. So I think that is definitely more important than the pure language you're trying to learn.
Yeah, I would agree. Along that line, what are some other programming languages that are commonly used for data science? I think we answered that. Python, I think, is the most common data science programming language. MATLAB is still huge, especially in industry for engineering. R is really big for statistical analysis in biology. There's Julia. C++ is still huge in anything that needs high performance computing. You're still going to need to rely on C. And Java is still big for anything that needs like nice visualizations, nice GUIs. Like Java has some really fantastic skills.
I took a Java class and I made a little house on a prairie.
Yeah. So I think that Python is the
SQL
biggest.
You count SQL as a coding language?
Oh, SQL is a coding language. Yeah, SQL is a different type of coding language, but SQL is really important if you do any databasing.
I think that's where you can pick it up pretty quick. So that I wouldn't I think that's where like this, if the situation calls for it, you'll be able to learn it. But it's not something to necessarily spend a ton of time doing.
I would also keep in mind a data scientist is different than a computer scientist. My job is not to be an expert in everything Python and how to do the exact perfect Python build. My job is to find results in scientific research, and I just need to know enough coding that I'm doing that legitimately. I can understand why I'm doing it, and I'm doing it efficiently enough for my purpose. I could always make it more efficient. I could always make it prettier, but that's not what I'm interested in. If I were, I would just be a software engineer and I would just do that all day. But I'm not. Okay, the next question is also on software, but Phuong this is probably more up your alley. What online courses would you recommend to learn ImageJ? And for that, I just say YouTube. just YouTube, whatever you want to learn for ImageJ, YouTube it. It's there.
Yes, YouTube it. I would say YouTube is the best. Although ImageJ does have like tutorials on, if you go to like ImageJ help or something, I think there are like small, like shorter tutorials that you can find, and they ImageJ also provides you some example images that you can like open up and like play with the different settings, like play around with ImageJ. But yeah, YouTube mostly for everything else.
I'll go into, I have one general one, but I want to end it with it. I have one specific one. Phuong, you had a specific one or no?
Yes.
Okay, so I'll go into my specific one. Someone asked me to explain microglia morphology live at least once. So microglia in a resting state are considered active resting, meaning that they're resting as they're not technically responding to disease or anything like that, but they have lots of branches. They're not super thick, and they're constantly moving throughout their environment to sense what's happening. There are so many ways they can change. They can completely retract all of those branches, which often happens during inflammation. It often happens during disease and injury. And in that case, they might also move into a phagocytic point where they can actually like cause blebs and engulf things and eat things, or they can even grow an end foot, so that literally looks like a little branch with a foot, and they can propel themselves to different regions of the brain that might be injured. Or they can keep those branches and they can actually grow on additional secondary and tertiary branches. So what we mean with that, if this is my arm, my fingers would be my secondary branches as they're coming off of my arm, and I can't do anything tertiary because that would add extra appendages. But imagine that there was another finger coming off of this finger, and that would be a tertiary branch. And then an additional thing that they can do is that they can be branched in just two directions. They can be branched around an entire circle, or they can even be branched, but the cell is more swollen. So the branches are thicker and the soma, or the cell body, is thicker. So they can just do a lot of things. And we don't really understand completely why they do one versus the other. And that's a big part of my research. So that's my specific one. Phuong?
Okay, my specific one is somebody asks, and actually several people asked me this, but I had one on the first module, the third module about, oh my gosh, no, the first module about immunohistochemistry. Several people were confused about whether there's a right answer. I asked what animal. I asked you all to name like a primary antibody as well as like the animal that the primary antibody comes from to stain like IBA, to stain like whatever target you want. And a lot of people are confused whether or not there's one single animal that's the answer. And the answer is no. Ideally, you would get an antibody that's specific to the animal that you are researching. So for example, for us, we use rats. So ideally, the antibody would work the best. You can find this stuff in the manufacturer like website when you're going and you're buying antibodies or you're researching about antibodies. It'll say it like in the paper or on the manufacturer website, what species the antibody works best with. Ideally, I remember Hawley. So for our work, we use primary antibodies derived from rabbits to stain rat samples.
In goat serum.
In goat serum.
Mm-hmm.
I know it is confusing. Watch the video if you're confused, but and it works fine. Even if we work with rats, we use rabbit antibody and it's fine. Mm-hmm. However, when Hawley used the rabbit antibody to stain an actual rabbit model, it actually works way better. Like the primary antibody, I believe, works way better.
It's because it's an anti-rabbit. It works really, really well.
Yeah, so ideally, that's what you would do.
We even got confused as well. So ideally, you're not supposed to pick an antibody that's in the same family. So if you're in a rat or a mouse, you probably don't want to pick an anti-mouse or the rat. But I had some sheep samples and the goat serum through both of us, where we're like, are sheeps and goats in close enough of a family that you can't use like a goat serum here? But apparently it was fine and it worked out just fine. But
you just have to try it out. Honestly, antibody staining. Mm-hmm. Staining is a process. There's a lot of optimization. You just have to find the one that works for you. But there's no correct animal. Like you just look for that on the manufacturer website or on the papers.
And likely you're going to be doing staining that other people have done. So just do what really works. And other people have tested it out. Yeah. So like it's pretty common in our field to do an IBA one with an anti-rabbit in goat serum. And so we'll just do that. Yeah. Cool. Other specific ones? Nels, do you have a specific one about MPT you want to throw in?
No, I don't think so.
Okay.
Everyone's like experts at MPT now. They have no questions.
Yeah. And Phuong, did you have another one?
No, I'm good actually.
Okay. So then our last one is back to a general one. And this is one that I've been getting a lot in the personal one-on-ones I've been doing with students this week, which is I'm still a bit of a loss for exactly what a high schooler or an undergrad does in a research lab. Why does the research lab need an underqualified candidate?
Not underqualified.
So, well, yeah, I've talked a lot. So Phuong, go ahead and start. You seem to have
just that you're not underqualified. Literally everyone starts somewhere. And the reason we just need help. Like I said, experiments, most of our time, I don't know if you have the poster there, but the scientific method, you cycle between experiment and analyze results. Most of your time is doing that. And so that's where extra hands can really help. Even if you haven't, even if you've never run immunohistochemistry before, I'll teach you. Like, a lot of it is on the job training. You just have to be there, show up and learn it. And
Also,
help me do it.
biological replicates and technical replicates. Like, I really should have had my undergrads imaging for me in my last
Oh,
6 months,
yeah.
but I didn't have anyone that was in a position. That those can be hundreds to thousands of hours of experiments. And so the way I was talking to it someone the other day was that if you come in as a high schooler, we understand where your level is at. We will train you on a methodology that aligns with your level. We'll give you the training, and you can still contribute quite significantly to our work while you're learning to get to the next level. And then in undergrad, we raise that level a little bit. You have a little bit more freedom, or your methodologies are slightly more complex. We can give you the training again. You can still contribute significantly to our research while PhD students are really pushing it, or PIs are designing like bigger ideas. And then, as a postdoc, it's raised again. And as a PI, that level is kind of raised again. So you're not underqualified. We're bringing you in for things we need help for at certain
We
levels.
just need hands and like brains. We just need extra time that we don't have.
And people with bachelor's degrees get full-time jobs as research assistants. That's how much, or research techs. That is how much research just needs to be done by someone that can learn a methodology and do it really well and do it a lot. And then when you get to a master's degree, you can be a research assistant, or you can be a lab manager. And then it's really PhDs that you become a scientist. That's literally, in a lot of companies, you're not considered a scientist unless you have a PhD. So even though you are a scientist, the job title doesn't become scientist until that level. So
yeah, definitely join a lab if you can.
And
whatever.
And just let them teach you. Don't try to be an expert. Even now, going into my postdoc lab, I know I'm not the expert. I'm gonna learn. I'm gonna bring in things that I'm an expert in. But yeah, closing comments, things that you wish people knew over the summer.
Just to reiterate what Hawley said, your job is just to enjoy life. Like, get to undergrad and enjoy life.
Yeah, do well in class. In undergrad, do well enough in class.
Yeah.
Don't even push for don't even do A pluses. That's too much work.
Oh, nobody has time for that. No,
yeah, nobody has time for that.
Nobody has time for that.
Nels?
Science is supposed to be fun. It should be you should do it enough where you enjoy it and you are learning, but it should also not be all consuming. So there's a balance that you should be striking. And also, if you are doing science and it's in an environment that isn't fun, you should consider changing to a different scientific environment. So you shouldn't have to push through unenjoyment or frustration.
Well, there's some frustration with it, but you shouldn't have to push through an unhealthy environment. Yeah. And then I guess I'll add to that, it's easy to compare yourselves to where the three of us are now, and that all three of us, Nels is almost at candidacy in his PhD. Phuong and I've graduated. Phuong's passed her candidacy. We've done a significant amount of research. But where we were at in your shoes, Nels and I had never done any scientific clubs. I didn't have any research experience until undergrad. Even then, it was pretty limited. I came into grad school with no papers. I only presented at ASU. That's fine. You're supposed to be learning now. Don't look at only the top students, and don't look at people that are 10 years in front of you and think that's where you need to be. Research is a long career path. You're not really going to start your career until your late 20s, early 30s. You're going to be in school for a long time if you choose to do an MD or a PhD. So just try to enjoy that process. Okay.
I also think it's good to every time I kind of hit another year, I guess, in school, think about how far I've come. And when I started my undergrad, when I was too scared to talk about anything, or when I just on the Nance lab where I didn't know what I was doing, and I was very scared to present, and I don't know, I still have a ways to go, but I really have grown. I think it can be really, really rewarding to think about how far you've come more than thinking about how far you have to go. You are truly going to just gain so much skills and confidence and knowledge just by continuing to do what you're doing every day.
Oh yeah, for sure. It's a journey.
It's
Yeah.
it is where you're in the long, if you choose research, it's a long haul type of career. It's not like athletics where you have to be the best by 25, otherwise your like body is old. You can do this until you're
It's old, oh no.
you can do this till you're 99. Take your time. Your thoughts will mature with time. All right. Thanks, everybody. I hope that this gave some more insight and let you show that the three of us sometimes talk to each other.
Yes, just sometimes.
Often.
Just sometimes. Bye, everyone.
Bye.